Hey, hi everyone. Uh, so we've been talking a lot about defects in our immune system. We've talked about how our immune system, while it is certainly very powerful and very finely tuned in order to be able to do a good job of detecting and protecting us against pathogenic threats that present different antigens that our immune system can recognize. The last few lectures, we've been talking about instances where you have defects in the immune system that might cause things like an immunodeficiency, where you don't have a certain component of your immune system working as strongly as it should. And that immunodeficiency could either be because of you're born with a particular genetic variation, a particular version of one of the many genes that code for components of your immune system, or the immunodeficiency could come about due to exposure to something in the environment, something that affects your immune system and causes the defect to happen, such as exposure to viruses like HIV. We also looked at how even a very strong, robust immune system can make mistakes from time to time. And so when we were talking about things like allergies, that was very much about how you have a healthy, normal immune system that for some reason makes a mistake, right, and begins to recognize some sort of benign, really quite harmless antigenic signal and interpreting that to be something like a threat. And therefore, your adaptive immune system might actually decide to spend time and effort making a immune memory against that particular antigen, so that if you're exposed to it again, you have a strong secondary immune response. And so we said harmless things that like pollen from flowers, or nuts like peanut oils, or certain types of other foods like fish, right, or seafood. We can get these types of allergic reactions to them. We also learned that much of that has to do with a sort of false calibration of our immune system. That is, I shared with you the hygiene hypothesis that says that as our modern lifestyles are getting cleaner, right, we are exposed to fewer and fewer types of pathogens in our daily lives. We are taking medications that either prevent us from getting sick in the first place, for example, vaccinations, so that you don't have to deal with the risk of developing some life-threatening infection from a virus, for example, right? But because we are depriving our developing immune systems of those types of encounters and those types of challenges, that maybe our immune systems are getting more and more sensitized to things around us that in reality they shouldn't be. And that's how we develop, allergies. Now, a couple of you asked me when we were talking about allergies, I kind of cautiously brought up that point about vaccinations being potentially part of this hygiene hypothesis, right? Because by protecting ourselves against different types of infectious diseases, are we in reality, not allowing our immune systems when we're kids to really get the type of exposure needed to fully learn and develop how to deal with them? And I said, yeah, but there's obviously two sides to that coin, right? Because the whole point of being vaccinated is to protect you against real life-threatening types of diseases, right? Even things like polio, right, or measles, smallpox, right? Or even COVID, of course. Right? We're dealing with our current sort of pandemic and our current sort of awareness of the need to try to get vaccinated. In other words, I would never say you should not get vaccinated just because somehow there's a possibility that it might make your immune system, less educated in terms of how to fight against diseases, because that would be ridiculous, right? That's like saying, you know, um, some child didn't get vaccinated against polio and as a result caught polio and now is paralyzed for life. But thank goodness, he can eat peanut butter because he's not allergic to peanuts, right? I mean, obviously, that's a very false way of trying to uh, get carried away, saying that vaccines might actually have a downside in terms of this hygiene hypothesis. We also noted how hypersensitivities, like allergies, play a very important role in things like the challenges that medical professionals face when trying to successfully do things like tissue transplantation. So we spent a lot of time talking about things like why blood transfusions, you have to match the ABO blood type, why during solid tissue transplantations, not only ABO blood type has to be matched, but also you have to take into consideration matching the HLA types, right? And again, I emphasized the very interesting thought that in reality, when you're doing things like tissue transplantation, you're going against what Mother Nature designed us to do, right? And I think it's kind of a weird thought, but I like bringing that up because it is interesting, right? Because we marvel at modern science, modern medicine that allows us to do things like save people's lives by transplanting organs. But at the same time, we have to acknowledge that the very act of doing that and trying to get away with that, trying to transplant an organ, is not something that Mother Nature designed us to do. And the opposite is true. Mother Nature designed our immune systems to reject that foreign tissue, right? Because our immune systems never would have had any reason as part of its training and education to expect something like that to happen, right? Tissue transplantations, especially solid tissue transplantations, are only something that we've learned how to do in the last maybe 75 years or so, right? Compared to hundreds of millions of years of our immune system being geared up and fine-tuned by Mother Nature, right, to be really good at what it does. So let's continue this discussion and now talk about the role of hypersensitivities in a different type of disease situation, autoimmunity. And very importantly, we'll make the distinction between autoimmunity and immunodeficiency. I have our class calendar open here because I wanted to remind myself to bring to your attention that here we are in week 15, hard to believe, right? So we will have 3 sets of new lectures this week, and also our very last lecture with new material will be next Monday. Right? Then next Wednesday, our class will be a review class for our 3rd exam, and then our 3rd exam will take place Monday, May 10th at 12 noon, which is the normal time that the final exam would be scheduled for our class. But in this case, it's not a cumulative final exam, right? It's just a 3rd exam, just like the ones we've had before, but we're doing it during our final exam time starting at noon on May 10th. So I wanted to remind you this week you do have a quiz. The quiz 12 will be on chapters 15 and 16. So it'll cover the stuff that we are learning today and also Wednesday. So as a result, I won't make the quiz available until after Wednesday's lecture, but I will give you until Friday night to turn it in, right? Because now that we're reaching the end of the semester, some things are getting a little packed in. Okay, so keep that in mind. Okay. All right, so let's talk about the role of hypersensitivities in autoimmunity. Remember that one of the fundamental principles of how our adaptive immune response works is that our immune system has to tell the difference between what types of signals are self signals, right? Stuff that comes normally from within our body, our normal healthy cells and tissues that present all kinds of 3-dimensional molecules that could potentially serve as antigens or signals that components of our adaptive immune system can pick up on. In particular, I'm talking about things like immunoglobulins, right, on B cells, and T cell receptors on T cells, that go through that whole elaborate process of VDJ recombination just to come up with so many different diverse shapes of binding molecules, IGs and TCRs, right? They have to be, as part of their training, told that you cannot bind normal signals that you find in the body. And we've already learned that in the process of B cells developing or T cells developing, right, any of those brand new cells that come up with shapes that bind to self signals, that leads to those developing cells failing self-tolerance training. Right? And such a B cell or such a T cell would not be allowed to go any further in becoming a fully functional lymphocyte as part of our immune system. Right? So self-tolerance training is really about exposing those developing immune cells, right, to plenty of opportunities for them to see what normal signals are present in the body. And then you eliminate any of those developing lymphocytes that improperly bind to those self signals. Okay. Every single day, our immune system, right, our body is exposed to many more examples of healthy, normal self-antigens than pathogenic antigens. Right? And I think that makes sense. I've mentioned that before, but I think it's worth thinking about because we think about our immune system. I think the average person on the street, if you ask them, what is our immune system dealing with every day, right? They'd probably say, oh, it's looking out for bad things that are coming in from the outside, right? Things that are on viruses or bacteria, and that's how your immune system knows to go ahead and react. But I think what the average person doesn't think about, and hopefully all of you are thinking about more and more as part of this course, is that every day, right, there are way more self-antigens that the B cells bump into or the T cells bump into, right? Many more of those self-antigens than there are pathogenic antigens. So here's a table that just summarizes some of the mechanisms that our immune system uses to help promote or encourage good, normal self-tolerance. So these are all things that we've learned before, but let's put it together in this table so that it helps us think about, when we move on to autoimmune disease, right, what we're talking about. So number one, for example, negative selection of B cells in the bone marrow. B cells that are developing in the bone marrow, they come up with their new IGs. If they're new IGs, those novel shapes end up binding to self-antigen in the bone marrow. That might be the end of the line for that developing B cell. Right? If it's lucky, maybe it can try to do VJ recombination a little bit more to come up with a new light chain of its IG so that the shape changes a little bit, right? And so that it no longer gets in trouble. But really the point is any developing B cell that fails central tolerance training in the bone marrow, that's the end of the line, right, for that B cell. Likewise, for T cells, we said that in the thymus, as thymocytes are learning how to make their T cell receptor, the thymus can present all kinds of tissue-specific proteins to give the developing T cell a wide range of exposure to different types of self-antigen, to make sure that that developing T cell doesn't auto-react against self-signals. Negative selection of T cells in the thymus, right? So remember, T cells undergo both negative and positive selection. One thing we didn't discuss is anatomically. There are certain types of tissues in your body, certain regions, where lymphocytes are kind of excluded from. Things like the brain, for example, right? Or the eye, right? So these are moves that are made to exclude lymphocytes from certain areas that might have very sensitive tissues and might not react well to T cells, for example, or lymphocytes overreacting. So obviously in your brain, you don't want to have a lot of B cells and T cells activating and causing inflammatory signals. Likewise, in the eyes, for example, you don't really think about this, but our eyes we're exposed to a lot of pathogens that are in the air, things like that, right? You want to carefully manage the types of immune responses that take place in your eyes, because your eyes, of course, are very delicate tissues. You don't want to do anything that ends up compromising the ability of your eyes to function. Uh, Autoreactive B and T cells are encouraged to undergo anergy, right? They're basically turned into just sort of deactivated cells rather than allowing them to continue. And we also mentioned that we have certain helper T cells, these Treg cells, that help keep the noise level of our immune responses down, right? And by doing so, it does so by discouraging the activation of other T cells and B cells as well that might be sort of approaching or visiting a particular target. All right, so those are all the ways that our adaptive immunity has this built-in system to manage self-tolerance. But what happens when this fails? When you've lost the ability to properly recognize and distinguish self signals from non-self. So that's what autoimmunity is. Autoimmunity is a failure of self-tolerance. Consider that if we routinely generated Igs or TCRs against our own body's antigens, right, our health would always be compromised, right? If you didn't have a way to self-tolerance train your lymphocytes, and any old B cell or any old T cell was just allowed to go to work, you would quite literally be ravaged every day from within, from your lymphocytes freaking out and activating against numerous self signals, right? And the toll that would take on your overall health, you can imagine, would be very severe. Right? If your immune system was always engaged and always battling your own tissues, right? And your body is somehow having to cope with that all the time, that's not good. Importantly, I want to make this distinction between autoimmunity and immunodeficiency, right? We've already learned about immunodeficiency, and I said the key here, right, is in the word. Immunodeficiency means you're missing something from your immune system, right? Either because of genetics, that you never had it in the first place, right? Or due to exposure, something caused a component of your immune system to be lost. Autoimmunity, on the other hand, right, has nothing to do with deficiencies. It basically is a perfectly normal, strong, complete immune system that has somehow made this terrible mistake now of recognizing self signals as being a threat. So you can already see some similarities with allergy, right? Except for allergy has to do with something from the outside that's not a self signal, right? Pollen or peanut oil or shellfish, right? Those are external things that are not part of your own body's tissues, and your immune system reacts against those. That is your classical allergy. But autoimmunity is when your immune system reacts against your own self tissues, right? And in many ways, that's obviously much more potentially dangerous and potentially debilitating. So to date, we know over 100 different autoimmune diseases that have been identified. And just like with allergies, right, they are more common in affluent, industrialized first world countries. Maybe that's not a surprise to you, considering, again, we're talking about immune systems making mistakes, right? And recognizing something as a threat that shouldn't be. So whether it's an allergen or an internal signal, right? Again, potentially the hygiene hypothesis might be sort of rearing its ugly head here. So the body then engages in what is essentially like a civil war, right? You have your immune system attacking your own body. And as a result, oftentimes, right, it's very hard to kind of figure out where the origin of this mistake is. Right? So even though we know over 100 different autoimmune diseases, there's really only a handful of them that we have a decent understanding of what causes them. What we do know, and what is most likely true for all autoimmune diseases, is that it is a combination of factors. Genetics plays a very important part, as well as developmental and environmental factors. So things like as you are brought up, right, what types of things you're exposed to. This includes not only things like infectious agents, but also good things like nutrition, right? This can all play a role potentially in development of autoimmune disease. All right, so this table in the book, this is a pretty nice table because what they've done is they've highlighted the fact that once again, what we are talking about are immune reactions that follow these hypersensitivity models, the same models that we introduced when we talked about allergens, the same models that we talked about when we were discussing grafts and transplantations, right? So type 2 hypersensitivity, type 3 hypersensitivity, and type 4 hypersensitivity. So just like those other cases of allergies or tissue transplantation rejection, type 2 involves antibodies against some sort of cell surface or matrix antigen, meaning the matrix of your tissue. Type 3 is the more chronic, drawn-out kind that involves immune complexes forming that your body has a hard time clearing. So again, antibodies interacting with different types of targets, causing large, maybe potential deposits that your body then experiences inflammation to. And then type 4, just like we discussed before, right? Type 4 doesn't have to do with antibodies. It has to do with T cells making a mistake. So this table lists some of the more well-known or high-profile types of autoimmune diseases. And some of these you probably recognize just from looking at it. Right? So in here you have things like myasthenia gravis, you have lupus, which is an autoimmune disease. You have diseases like multiple sclerosis or type 1 diabetes, right? Some of these, you might be surprised. Right? I think lupus, you guys probably knew something about as probably having to do with your immune system, right? But type 1 diabetes, did you know that that has to do with your immune system? Or things like rheumatoid arthritis. Right? So what we'll do today is I'll take you through one example of each of these, type 2, type 3, type 4, so you get an understanding of what's going on. And then next time, we'll maybe finish talking about the examples on this table and then wrap up our discussion about autoimmunity. All right, so let's start off just taking one example from each. So autoimmune hemolytic anemia as a type 2, systematic lupus erythematosus as a type 3, and MS, multiple sclerosis, as a type 4. All right, so let's look at these. So hemolytic anemia. The name itself gives you a clue, right? It is anemia. So anemia means what? A compromised ability of your blood to be able to carry enough oxygen in it, right? And so this has to do with your red blood cells. Hemolytic gives you another clue, right? Lytic meaning lysis or breaking. So this is the breaking or the lysis of your red blood cells, right, due to an autoimmune reaction. So I think we've already discussed enough about ABO blood typing and the pitfalls of ABO mismatching for you to already understand or guess what type of autoimmune mechanism is going on here, right? This is when your body makes the terrible mistake of selecting for B cells that have Igs on their surface that bind to your own ABO blood type. So here you see this red blood cell, right? It has these blue antibodies that should not be binding to the ABO blood type here. So the E stands for erythrocyte, right? Red blood cell. And as a result, what can this lead to? Well, what does the coding of a target with antibodies lead to, right? It leads to things like macrophages suddenly taking an interest in eating your red blood cells, right? So now the macrophages with their Fc gamma receptors are holding on to the stems of the IgG. As a result, phagocytosing and destroying red blood cells. Remember, the attachment of IgG onto the surface of a target also results in opsonization with complement proteins, right? Because complement proteins are attracted by antibodies. And as a result, the macrophages might also start binding these red blood cells because of their complement protein receptors, their CR1 receptors, right? I mean, that's kind of terrifying, because starting all the way back at the beginning of the semester, when we used the example of the beach ball macrophage attacking the little ping pong ball bacteria, we learned that the whole point is that things that make the ping pong ball easier for the macrophage to hang on to, right, is a powerful mechanism for getting your innate immune system to be able to attack a target that otherwise was having a hard time eliminating. So here, unfortunately, the red blob is no longer an invading bacteria, right? What's really terrifying about this is your own red blood cells being destroyed, right, by your own macrophages. As well, we also learned that if you start attracting complement proteins, you can end up initiating the membrane attack complex. So you end up poking your red blood cells full of holes, right? And as a result, they lyse. Right? So obviously, this is a big mistake of the immune system, right? And it's a big mistake that if it keeps going, will have disastrous results for the ability of the individual to maintain a normal, healthy lifestyle. Type 3 immune diseases involve immune complexes forming. So sort of a classic example of an autoimmune type 3 disease is lupus. Right? So lupus, again, has to do with the generation of IgGs improperly. But in this case, it's not necessarily directed against ABO blood type antigens. In this case, the IgG has been improperly selected for binding against a variety of different types of proteins in the body. Okay? The important thing to keep in mind is any of these potential protein targets are just normal everyday types of targets. Right? They can be proteins. A lot of times they can be other molecules as well. Things like even DNA itself can be improperly become a target of IgG production. And as a result, because these antigens are so abundant throughout your tissues, you're going to get a lot of cross-linking of IgGs bound to things that are then cross-linking with each other, and you'd get essentially clumps, right, or complexes that have to be then cleared away by cells like macrophages that go around cleaning up cellular debris. But if you have so much of this happening, right, then you're just going to have buildup of these complexes throughout the different tissues of your body, and that is going to trigger inflammation, right? Pretty severe inflammation. This can lead to heart attack, stroke, organ failure if the capillaries of different tissues of organs like kidneys end up being clogged up with all these immune complexes. Okay? This is an interesting point and an important point when we talk about autoimmune diseases. 90% of lupus patients are actually women between the ages of 15 and 44 years old, right? That is really striking, right? So 9 out of 10 lupus patients are female. Currently, we don't have a cure for it, but fortunately, because the most pronounced symptoms are inflammation, you can treat lupus patients with anti-inflammatory drugs. Right? So the symbol for lupus, lupus, recognition is the purple ribbon. And oftentimes the butterfly is used as a symbol because one of the common symptoms is inflammation that manifests as a rash over the face in this kind of butterfly-like pattern. Right. I think lupus is something that a lot of people were sort of marginally familiar with because you hear stories about people getting lupus. I think it wasn't until maybe 5 years ago or so when pop star Selena came public with her own battles against lupus that lupus was really kind of brought to the forefront of popular public awareness. Right. So in her case, her kidneys were failing due to her lupus, and she had a much publicized kidney transplant, right, from her good friend. Type 4 autoimmune diseases. Type 4 autoimmune diseases don't involve antibodies, but just like other type 4 models, right, and allergies and graft rejection, they involve T cells. So multiple sclerosis is a classic example of a type 4 autoimmune disease. Multiple sclerosis, or MS, has to do with the degeneration or the destruction of nerve cells. So nerve cells, as pictured here, consist of a body of a cell with lots of dendrites where it receives signals, and then a long kind of chain part, which we call the axon. And that's the part of the neuron that's able to transmit these signals so that it can then communicate with another neuron, or even with something like your skeletal muscles, so your muscles can move in response. So this main axon region is surrounded by other cells that basically coat and insulate the axon part of this cell, right? So that you have these cells that create what we call these myelin sheaths. That's why oftentimes when nerve cells are drawn in a cartoon, the axon part kind of looks like, a length of sausage links, right? Because the reality is each one of these sausages is actually a myelin cell wrapped around the axon to provide insulation. Why is insulation so important? It's just like the insulation of wires in your home. When you have a wire that, let's say, a cable that's plugged into an electrical outlet that runs electricity to some device, whether it's a lamp or some other electronic device, right? The wire is always insulated, has insulation on it. That's not only to protect you against getting zapped when you touch the wire, but wires that are insulated allow the electrical signal to move much more efficiently. Right? So Mother Nature discovered this a long time ago, before humans ever discovered electricity. That's why the axons of our myelin, of our nerves are covered in these myelin sheaths. You can see here, when you have proper coating of the axon and the myelin sheath is intact, the message, the signal moving down the axon, moves very quickly. Right? They estimate that an electrical signal moving through an axon in your body can move at about 400 kilometers per hour. Right? However, if the myelin is damaged and you lose that insulation, then you can slow that signal down by a factor of 100. So if you think about this normal healthy myelin coated axon might be running from, let's say, your spine all the way down to your foot, right? And if you think about how long does it take for you to have the thought of, okay, I'm going to move my foot, to the time your foot actually moves, we think of it almost as instantaneous, right? That's how quickly the messages can get down there. But if you were to slow that down 100-fold, then you can imagine, right? You literally have a lag between the ability of your brain to send messages down to your muscles, and then your muscles actually responding in a much more delayed fashion. So in this case, this destruction of the myelin sheath is due to T cells making a mistake. So in this case, Th1 CD4 cells activate against the myelin sheaths of the neurons, right? Because remember, T cells bind to other cells and decide what to do with those cells. Right? In this case, the CD4 helper cells think that the myelin cells, right, are some sort of threat. And as a result, there's a release of cytokines like interferon gamma that recruits macrophages, and the macrophages begin to destroy the myelin. As a result, you end up with these scar-like plaques, or what we call sclera, right? And these kind of scarred regions of the neurons really impede the ability of the nerve to conduct. So some of the symptoms are loss of sensory skills, muscle weakness, a loss of coordination. Okay? And so MS, so this is multiple sclerosis, right? Because you have multiple of these scar-like sclera all through your nerves, right? It usually presents between the age of 20 and 50. And again, patients are more likely to be women than men. In this case, twice as likely to be women than men. And then the color of the ribbon for recognizing MS is orange. Right. T cell training can also play a big part in this problem. Remember how brand new T cells that are forming in the thymus are presented with many, many examples of potential peptide signals that would be found in tissues from throughout the body by the thymus cells, kind of just expressing little portions or bits of these peptides for the developing T cell to get a preview of. And we said that that ability, that really remarkable ability for the thymus epithelial cells to express random peptides from different tissues all over the body, has to do with this transcription factor known as AIR. So AIR is that transcription factor that within those thymus cells goes around just kind of randomly turning on different genes so that you express little peptides and things like that. AIR, of course, itself is a protein, and that means the AIR protein comes from the gene that codes for AIR. Right? So if you have defects in the AIR gene, okay, so this can be hereditary, then it may result in limited ability of your thymus epithelial cells to really express or show a diverse range of different tissue examples of peptides to your developing T cells. And as a result, the developing T cells don't properly get trained to recognize self signals. And you may have T cells that are then allowed to go to work in your body that should not have been, because they might end up being very good at detecting a self signal somewhere else in your body and viewing it as a threat. So here on the bottom is a table from a European study. All right, so this is a frequency of Finnish patients who are suffering from this disease, APD, or autoimmune polyglandular disease, right, that results from having a defect in the AIR transcription factor. And you can see among these patients, many of them secondarily suffer from different types of symptoms having to do with their T cells not being properly trained. So things like hypoparathyroidism, so the parathyroid gland, right, being attacked by T cells, right? 85% of these patients show signs of that. Other types of failure, like adrenal failure, ovarian failure, even the development of things like insulin, and other types of infection. So candidiasis, right? Candida is different types of yeast that can cause yeast infections, right? And you can see 100% of these APD patients suffer from out of control yeast infections, right? Et cetera, et cetera, et cetera. All from improper or inadequate training of their T cells during T cell development. So much of this then requires us to think about these mistakes that are being made by our immune system. In other words, detecting things that T cells normally, for example, should not be allowed to detect, right? That must have a lot to do with how well certain types of potentially problematic peptides are actually being presented to an immune system that is more likely to make a mistake, right? Because remember, a T cell with its T cell receptor, it's not going to make a mistake against something that it never sees, right? But it's only when a cell presents something, then there is the opportunity for the T cell to potentially make a mistake, right? Because it has a T cell receptor that binds that thing. So this gets back then to some genetics here, right? Because if your cells of your body are less likely to present certain types of problematic peptides, even though they're normal peptides, right? But certain peptides might be more prone to having your T cells make mistakes against. If your genetic makeup means your cells don't present those potential problem-causing peptides as well as somebody else's does, then maybe in reality, you have a less risk or a lower chance of your immune system and your T cells making a mistake. Right? So this goes back to HLA polymorphisms. Right? So remember, here's the major histocompatibility complex on chromosome 6. So this is the gray thing, of course, is a physical length of DNA, right? Part of chromosome 6 that we're zoomed in on here. And in this region, you have the class 1 HLA genes, so HLA A, B, C, E, F, and G, right? And over here, you have the class 2 HLA-D genes, right? And remember, these are the ones that then make HLA A, B, C, E, F, G, that make our MHC class 1 platforms, and the class 2 HLA-D genes, right, code for different variations in the MHC2 platforms. Right? And some of these, again, are much more polymorphic than others. Right? So the ones in red here, HLA A, B, and C, so those would be these genes over here, A, B, and C, right? They tend to have far more variations in different genetic sequences in them in the human population compared to other of these genes. For example, HLA-F, right? This gene over here is monomorphic, right? There's really only this one genetic sequence that you see in human beings. So let's try to tie this together here, right? Because these different types of HLA proteins can be very polymorphic, right? We've already learned that the reality is, even though, for example, something like HLA-A, right, or HLA-B, where you might have over 1,000 different genetic variations of these that have been cataloged in the human population, it's not like every single one of 1,500 different possible polymorphisms is equally represented in the population, right? In fact, the reality is only something like 3, maybe 4 out of, a whole bunch of different possible genetic variations that are seen in people, right, are actually the ones that most people have. So for HLA-A, for example, if you look at this pie chart here, right, if this was meant to represent all like 1,000+ different possible genetic variations that are seen in human beings, right, the majority of them are this blue one, this brown one, or this green one, followed by maybe this purple one, right? And then the other 900+ different ones are so rare, right, that they don't even show up well on this pie chart. The way they name these polymorphisms, the way they catalog them, is with this kind of confusing nomenclature. Right? So for example, they use this system of an asterisk, right, and then the main polymorphism type that you're talking about, and within that sort of a sub-ID number identifying what version of the polymorphism you'd find, right? So you'd end up with something like HLA-A, right, the gene for HLA-A, and this particular polymorphism, O2:04. So if you look over here, right, here we have maybe the top 6 most common allotypes for this HLA, right? And for example, the 3 most common ones for HLA-A are the A2, right, A24, and A68 variants, right, with these sub sort of like details in there. I don't expect you to memorize these, because I don't even bother memorizing. There's no point in memorizing these, because these are all things you would just look up if you were actually doing this type of research and following this kind of stuff. But I do want you to be able to at least recognize that when you see nomenclature like this, that's what they're referring to. Okay. All right. Why is this relevant to autoimmune disease? Because, as we were just saying, depending upon the shape of your particular HLA protein, right, due to the genetic variation that you have, right, that means like 1 or 2 amino acids might be different. So the shape of that particular protein is slightly different, right? And that HLA is what makes your MHC platform. If the purpose of the MHC platform is to be able to hang on to and present peptide on the surface of a cell, then yeah, depending upon your particular polymorphic makeup, right, you may or may not have a variation of that shape that's particularly good at hanging on to a particular type of peptide and presenting it. In fact, your body, right, due to your ancestry and the type of polymorphisms that you have, maybe the shape of your MHC platforms are really good at hanging on to a particular type of peptide. So your cells show this peptide off a lot to your immune system. On the other hand, maybe your genetic makeup is such that the shape of your MHC, right, is really bad at being able to hang on to a particular peptide. And as a result, on the surfaces of your cells, you don't show that particular type of peptide, very much, if at all, to your immune system, right? And if it's the presentation of that peptide that is actually providing the opportunity or setting the stage for your immune cells to make a mistake in the first place, then hopefully you can see what I'm getting at, right? Depending upon your genetic makeup, you might have elevated risk of developing a particular type of autoimmune mistake, or you might actually have protection, much lower risk than normal of your immune system making that same mistake. So here's some examples. Keep in mind, these genes that make our HLA proteins that are part of chromosome 6, right, are physically close to each other on the chromosome. That means they are inherited as a block of genes, right? So during meiosis, right, during the formation of gametes, when there's crossing over, that allows genetic mixture and recombination to happen, right? Because the recombination is a physical recombining of chunks of chromosome arms that swap over between the one that you got from dad and the one that got from mom, right? They may swap over. Blocks of genes are inherited together. They're linked together. Right? So that's what we call a haplotype. So here you can see there are a number of different allotypes here, right? Meaning what particular set of polymorphisms, right, that are associated with having increased risk of diseases. So the HLA-B27 polymorphism or allotype, all right, is highly associated with this particular disease, ankylosing spondylitis. Here are some other ones. For example, celiac disease, right, has to do with what genetic polymorphism of HLA-D you have. If you have the DQ2 and DQ8, right, you would see that 95% of patients who suffer from celiac disease, there's evidence that they've inherited this particular polymorphism in their HLA-D, so on and so forth. That means there are certain blocks or certain sets of these genes, because they are inherited in chunks or haplotypes, that together as a haplotype block are more associated with different types of autoimmune disease susceptibility. So in particular, this one here, if you've gotten the HLA-A1, B8, DR3, DQ2 combination, right, that means you inherited this variation or the set of variations, right, you have a much more increased risk of developing certain autoimmune diseases. All right. So I'm going to end with this slide today, giving you an example of celiac disease to really highlight what I'm talking about when I say that this genetic component is hard to ignore. So celiac disease, it can be a very severe condition whereby people don't just have gluten intolerance, but their sensitivity to gluten is so severe that they can actually get really, really sick, right? And their health can take a real dive if they end up eating foods that have gluten in them. So gluten is a protein that's found in wheat, right? And gluten is rich in the amino acid glutamine. That's why glutamine, the amino acid, is called glutamine, right? Because it was originally discovered and extracted from gluten proteins. So in the cells, so if you eat, for example, bread that has a lot of gluten in it, in your digestive tract, it's broken down, and a lot of these glutamine or these small little peptides or pieces of gluten protein, right, get processed by your cells and get presented on the outside of the cells of your GI tract. Right? Now, in the cells, these get converted to glutamate, peptides that are rich in glutamate, which are negatively charged amino acids. So that means you have these peptides that are very unusually negatively charged. And as a result, MHC platform shapes that are good at hanging on to negatively charged peptides would do a better job of presenting these peptides on the outside of your intestinal cells, right? So what they found is that people who have particular polymorphisms, the DQ2, you can see here, if you have the B1 star O2 or the star O5 or the DQ8 O3 if you have this particular combination, that means the shape of your MHC proteins, right, are really good at hanging onto and presenting these negatively charged peptides that come from the gluten protein. That means your chances of your immune system making a mistake against this peptide is much higher than somebody who, for example, right, if you don't have this particular combination at all, and your cells of your intestines are really bad at showing off these negatively charged gluten peptides, then there's a very low chance that your immune cells will ever see them and make a mistake against them, right? So you can see here, right, depending upon the particular genetic composition you can have anywhere from very low to very high risk of developing celiac disease, just as part of your genetic makeup. And again, it's interesting to think, right? It all has to do with how well your cells are presenting a particular signal that your immune system, right, must see in order to potentially make a mistake in the first place when it sees it. And if your cells don't show it, then your immune system never sees it and never makes a mistake against it. Okay? So you can see here, this is the normal lining of the intestinal tract. These are of the jejunum, these are like the cilia, right, that absorb nutrients and stuff like that. In this case, what's happening is T cells are reacting against the gluten peptides that are presented on the outside of the epithelial cells. And as a result, cytotoxic T cells come and start killing your intestinal lining cells. So instead of having these nicely formed epithelial columns here, right, basically the epithelial cells are just ravaged by your own immune system. And as a result, you cannot absorb nutrients, you have inflammation and other types of debilitating side effects. But importantly, this is where celiac disease and gluten intolerance is very different than other traditional or conventional autoimmune diseases like the ones that we talked about today. Why? In those cases, right, you don't have an opportunity to really intervene and try to eliminate the cause of the problem. But with celiac disease, you do. Because remember, the whole problem here is caused by the consumption of gluten proteins that give you the gluten peptides that are being shown off to the immune system, and the immune system is freaking out and attacking those healthy intestinal cells that are showing these peptides, right? So all you have to do is stop eating gluten. Once you stop eating gluten, right, the cells stop displaying the problematic peptide, right? And as a result, then hopefully over time, your symptoms go away. And just as long as you stay on a gluten restrictive diet, right, you can actually live with this disease and have a normal lifestyle, right? So that's why this is different. Celiac disease is different than normal autoimmune diseases, because in this case, right, you can actually easily, well, relatively speaking, easily adjust your lifestyle to simply avoid the gluten protein, right? So in some ways, this almost makes it more like an allergy, right? Because we talked about avoidance of allergens as being a primary way of dealing with allergic reactions. Okay. All right, so I'll stop there. And next time we'll continue looking at other examples, right? And hopefully you guys find this very interesting. And hopefully this gets you guys thinking about this whole idea of the genetic component, right? The genetic component that oftentimes has to do with how well your cells actually end up presenting things that cause the trouble in the first place, right? Okay, stop.